Sample swaps, contamination, and mislabeling are major issues in large-scale DNA sequencing studies, where a single mix-up can erase the discovery and diagnostic potential of thousands of samples. Quality control methods do exist, but they require jointly called germline variants, which prevents their use in cancer projects which only have somatic calls between tumor-normal pairs. Also, jointly calling variants in large cohorts can be very expensive and may need to be rerun if problems are detected. Somalier is a new method that avoids these problems by operating directly on alignment file. With Somalier, mix-ups can be identified before variants are called and with any sequencing experiment, including cancer studies. Somalier extracts sample sketches from BAMs or CRAMs and uses them to evaluate relatedness and other quality metrics. Here is a Somalier report for a cohort of 1,000 samples that ran in about 4 seconds. On the right, each point is a sample and the color indicates the sex given in the project manifest. The current axes compare the specified sex of each sample to the sample's X chromosome sequencing profile, with the X giving the number of homozygous alternate alleles, and the Y is the scaled mean coverage. As expected, female-labeled samples cluster with a normalized depth of 2, and male-labeled samples cluster at 1. Male-labeled samples also have more homozygous alternate alleles on the X. New plots can be dynamically generated by changing the axis values from the drop-down boxes. For example, another good general quality control check is to compare the proportion of sites with an allele balance outside of 0.2 to 0.8 to the mean depth of genotyped sites. Samples with a high value on the X are generally of lower quality, though there are no extreme outliers in this cohort. Also, we can see that most of these samples were sequenced to around 25 to 30X. There are many other options to choose from. The number of known sites can also be informative. If we hover over a point, it gives metadata about the sample and highlights a number of points in the left-hand plot. In that left plot, each point is a pair of samples that are again colored by information given in the manifest. Here, the color gives the relationship between samples. The current axes compare identity by state values. IBS 0 on the X is the count of sites between a pair of samples where one sample was homozygous reference and the other was a homozygous alternate. This should not happen between a parent and child except in the case of a deletion. And so we see that the expected parent-child pairs all cluster close to 0. IBS 2 on the Y-axis is the count of sites where a pair of samples had the same genotype. The cluster above 16K are duplicate samples and identical twins. This is a cluster of siblings. By switching the y-axis to relatedness, we can see that the siblings have a relatedness of around 0.5 and an IBS 0 above 0, as we would expect. We can also see that the duplicate samples have a relatedness value of 1, again, as expected. When we hover over a point in this left plot, the pair of samples on the right hand is also highlighted. This is a well-behaved cohort, but we do have a few samples without a sex specified. There are also quite a number of parent-child pairs that have not been specified in the manifest. Since Somalier can run on this 1,000 sample dataset in 4 seconds, it's simple to adjust the manifest, rerun, and get the most out of our data.